So we've talked about DNA structure. What else have we talked about thus far? So we know where DNA came from. We know the forms of DNA. And all this is in the review. I'm not going to go back over it. But there's still some questions. What does DNA really do? And that's a big question that's been answered yet. And so what does DNA do? And the next big question is, when and how is it replicated? All right, so that's another big question. And how does a cell divide?
We do
Mitosis, semiosis, yeah. Tomorrow we're going to do a virtual lab. Oh wait, today's Tuesday. I normally would have done a virtual lab today, but I had a meeting. And yesterday I missed, so we're going to skip the virtual lab this week. But there will be a lab. We're going to pull out the microscopes and we're going to go do a lab on mitosis, semiosis on something called onion root tips. We'll look at that. So how does a cell divide? That's another question. That's a big question. And so to answer all this, we're going to cover some topics, all right? So it's going to be a lot, but I think you can handle it. We're going to do DNA replication. Bless you. I want to emphasize to you again one more time that our test is in April, and so we have to push this stuff. DNA replication, right? We have another big thing we're going to cover is mitosis and meiosis. And another big topic we're going to cover is the cell cycle. And the last big topic, and I say big, it's big, transcription.
With translation.
And translation. And so each one of these covers a major idea of what and whys. So why is DNA important? What does DNA do? It's this, transcription and translation. It's understanding how things are made and the fact that this actually will help you understand what a gene really is. And once we go through all this, and I have an activity for you guys to actually make an RNA molecule and make a protein, and you're going to do that in class, or maybe for homework. But then over Christmas, over the winter break, what I want you guys to do is start to go over some more complicated Mendelian genetics. I think you can handle it. It's going to be challenging, but when we come back, as we've done before, when we come back, we'll go over the answers. I might even send you a video on it, okay? All right, so transcription, translation. Let's start off with this idea of DNA replication. Does anybody know what do we know about DNA, making copies of DNA? What do we know about DNA replication?
It starts off with
We got 4 sequences.
it starts with
They start, guys, give me, yeah, I hear some answers, but they're not very confident. Give me, it's semi-conservative. That's for sure we know, right? So for sure, without a doubt, we know this, that DNA is it's replicated in a semi-conservative manner. Everybody understands that. Well, that means that you're making copies, you're going to have one old and one new. We know that from the what experiment? Messelson-Stahl. Good, Messelson-Stahl. So excellent. No, that's good. That's good enough, as long as you're close. All right, so what we have then is a DNA molecule. I'm badly drawn there. Okay, so there's a DNA molecule. And there's going to be some vocabulary words here you have to kind of try to get in your mind. The one thing you have to understand about DNA, it's strange, and you'll have to think about let me pause this for a second. So this DNA molecule has polarity, and what it is that you have a 5' end, 5', that's what we call that little the hyphen, right, or the apostrophe, okay? It's 5', and over here you call it 3'. Now, and down here at the opposite end is the other. So one side's 3, and for each molecule of DNA in the chromosome, you have a 5' end and a 3' end. Is that okay with everyone? So you see they're opposites, right? You see they're opposites, like wherever you see a 5, you see a 3. Wherever you see an A, you see a T, right? Because of that, we call this anti-parallel. Did I get the spelling right? Anti-parallel, okay? So they're parallel, but they're opposite, so we call them anti. All right, that's what anti means, right? Opposite. So that's part number 1. The second thing is it's 3' and 5'. Let's look at it for just a second, and let's remember that what are the 3 parts of a nucleotide? This is where that work we did. That's right, a pentose sugar, excellent.
A phosphate.
Excellent. So we got a pentose sugar. Lovely, perfect. So this pentose sugar is numbered. All right, so pentose, what does that mean? How many carbons? 5. So here's 1, right? Remember, wherever the lines come together, we call them 1. And there's a line sticking up here. 2, 3, 4, and 5. This is 1', 2', 3', 4', 5'. Is everybody okay with that?
Yep.
So that's what this 5' means, that this is this part sticking out, and the 3' means it's this carbon down here. Is everybody okay with that?
Yep.
All right. So that's where the 5' and 3' come in. Now to draw a DNA molecule, I'm going to use NB for nitrogenous base. Remember, we got pyrimidines and purines, right? We have double. Am I recording? No, not double bonds, but I'm blanking out here.
Polyx.
No. Anyways, geez, what the heck is I couldn't remember rings. I think it's circles. I know it's not circles, so okay. It's double ring structures or single ring structures, okay? And so remember that, right? So one type of, you have ATGC, right? huh?
The nitrogenous bases.
So there's pyrimidines and purines, right?
Purines is C and T.
Is it C and T? Which one's, it doesn't, you know what, I hate to waste our time on this.
The longest,
It's not, say what?
the longest word is a CT. CT.
Cytosine, thymine, adenine, guanine.
Okay, so the pyrimidines, all the pyrimidines have a single ring. So they look like this. 6-ring structure. They're nitrogenous bases, so they actually have a nitrogen in it here, and that's and there's a nitrogen over here. Remember, that's how Messelson and Stahl were able to use the fact that there's nitrogen in these rings to identify how DNA was replicated. So that's one type of ring. And there's double bonds here, guys, and there's all kinds of structures I'm leaving out. There's some double bonds here. Then you have the purine. The purine's actually 2 rings. I can't believe I forgot ring and just kept thinking circle for some reason. My brain's not working as well as it used to. I'm getting old, I guess. All right, so there's nitrogen in here as well, all over the place. And again, the Messelson and Stahl were able to use this fact as a way to identify the structure. So these are the purines, and the pyrimidines are single ringed. And the pyrimidines the single ringed ones are C, U, and T. Those are your,
Oh, it's a U.
U is in RNA, right? And that's another thing we'll learn in just a minute. So cut is for pyrimidines, and A and G are for the purines. Do you really need to know the difference between purines and pyrimidines? At this stage, no, I don't think they're going to test you on it at the end of course exam. But you should know pyrimidines. How do you spell pyrimidines?
P-Y-R.
P-Y-R-I-M-I-D-I-N-E. And I ran a space. Oh, I can't. Okay, there it is. That's fine. I'm writing with my finger. My pencil's getting charged. I apologize for that. And the other one's purine.
Well, it don't matter.
So am I going to ask you a question to identify purine versus pyrimidine? Absolutely not. This is just a classification of the different nitrogenous bases. But I'm not going to really concern myself with that right now. I'm not going to ask you to identify a nitrogenous base on the test. I don't think they're going to do that to you on the end of course exam. So, but it's important to have some kind of concept of what's going on here. So I'm not going to worry about that for right now, but you never know what's going to happen on a test like this one you're facing in April. So I'm going to worry about the strand, all right, one DNA molecule. And what you'll find is you have a phosphate with double bonded to an oxygen. You have a phosphate that's going to be double bonded in phosphorus is going to be double bonded in oxygen, single bonded in oxygen. There's going to be a negative charge. Single bonded in oxygen will be a negative charge. So that leaves you open to make some bonds, right? You take those and make a bond. Then you have an oxygen, and then that oxygen is going to be bonded to a carbon. And I'm obviously going to have to make this smaller. So an oxygen is going to be bonded to a carbon. Then it's going to be bonded to another carbon that's part of the ring, the ribose ring, the pentose. So what carbon is that I just drew out? It is a pentose, but what carbon, what number is it? Always start at the 0 and go right. So this is 1 prime, 2, that's right, it's the 5 prime, 2, 3, 4, and 5. So this is the 5 prime carbon. On the 5 prime carbon, what do you find? Phosphate. It's from the first prime that you get your nitrogenous base. So I'm going to put nitrogenous base, and I'm going to put I'm just going to put a circle because it could be either pyrimidine or purine, and it's going to be bonded to that 1. Okay, so the one carbon is where you find the nitrogenous base, but you need a chain, don't you? You need to have a chain, and so the 2 that you have to worry about for the chain is the 5 and 3, because this 3 is going to be bonded to what? What do you think? To close, very close, to a phosphate, which means to an oxygen, to phosphorus, double bond to oxygen. There's still one little negative, and that negative is why DNA is charged, right? DNA is polar because of this stuff. It's charged and polar. And you have an oxygen. But that phosphate, though, this phosphate is going to be connected to what? Somebody said it already? That 3 prime is going to be connected to a 5 prime, that's right. And that 5 prime is connected to the ribose sugar.
So it keeps going.
And it keeps going for a very long time. Billions, literally billions long, right? And I'm not going to be drawing that. But this is, again, 1, 2, 3, 4, 5 carbons. Those are the 5 carbons of your pentose. And then you get your phosphate, and it's going to keep going forever. But on the other end, at the very end of this, okay, what are you going to get? What's going to be on this end? One end is the 5 prime, and the other end is going to be a 3 prime. Do you see that?
Yeah.
Because it's going to be, if this was, wherever it ends, the last one is going to be like this one, where you got what? A 3 prime. That 3 prime, I should say, has this thing called a hydroxyl group on it. So it's an OH. The 5 prime has a phosphate, and the 3 prime end has a OH, or hydroxide. It's part of water, right, but it's hydroxide. Yeah.
So why is that 3 got phosphate on it? Actually, the first one. We do another phosphate.
Well, if you're going to put another nucleotide, what would you attach to this hydroxyl group? You'd attach a phosphate. You'd probably end up making water, right? So it's called dehydration synthesis, right? So when you're coming and making a polymer, if you were going to add another one, you'd add it here, you'd add a phosphate to it, you kick out water. So you see that if you look at it, that's what, this used to be a hydroxyl group here, but then phosphate came in and did what? It connected to it and kicked out water, right? And so if we were able to go back, you can go back and rewind the video and look just at the previous slide, you'd find that this structure has a ribose sugar, a nitrogenous base, and a phosphate. And that's what you're adding one to another, linking them one group at a time. Are we good with that? Okay, so this is not, that's the basics of it. And of course, if you have on this side a phosphate, and it's connected to the 5 prime, and that's coming off the sugar, as we said, and that's an oxygen, and it's badly drawn, I know, and there's a nitrogenous base here. If we have this going on, what do you think is going to happen on the other side when we see this nitrogenous base has to be lined up so that it can make either double or triple bond with the one on the other side. And so the nitrogenous bases have to line up. And because of the way they have to line up, what you get is an opposite, the anti-parallel. So instead of having the 5 prime on this end, you're going to have the 3 prime, which means you're going to have it upside down. It's going to be anti-parallel. It's going to be parallel, but it's going to be the opposite direction. So you're going to have the 3 prime here, and it's kind of hard to draw. I'm trying to draw it correctly. Give me a second. So we have this is connected to the 3 prime. So you don't have
Is there a 1 and 2 prime?
So there is. I showed you a minute ago. You can rewind the video later and look at it and and answer that question for yourself. But there is, and you have a phosphate here, and it looks crazy. You don't have to know. I'm not going to ask you to draw it, but what I'm trying to get through to you is that this is all upside down, and so you have here hydroxide. See?
Right.
So here you would have, on this side, you normally would have a hydroxide. Do you see how they're upside down?
Yeah.
So that's why we call it anti-parallel, that's all. So if we were going to add another phosphate, we'd add it here, right?
Right.
And this side has to add it phosphate here, then phosphate, then phosphate. Here's phosphate, and then phosphate. But hydroxide is at the top here. So we call this the 3 prime end. And what's this side?
5.
So you see how they're opposites. And then when we get to the end, the other end, this side will be 5 prime, right? And this will be 3 prime. Does that make sense to you?
So
This is, the anti-parallel is that when these two molecules, these two long lines, what are they? Are they parallel to one another? Yes. One's next, they're parallel like this, right? One's next to the other. But it's anti because one's upside down.
Right.
Does that make sense?
Yeah.
So they're parallel but upside down, so that's why we call them anti-parallel. And that's just the way it is. Because of the tree of the molecules and because of the chemistry, they have to be upside down. So now comes what a lot of people consider the hard part. All right, now we're going to get into the hard part, so try to figure out. All right, go ahead. Go ahead, someone asked a question.
So will you ever get like a sequence where it'll be 1 prime on top, 2 prime, or something like that? Will it?
No, this is always the way it works. It's the chemistry of it. It doesn't work any other way. It literally can't work any other way. So you kind of need to get that in your mind as we're going along, that what you need is you need to have, they always are anti-parallel. They're always 5' then 3'. I mean, that's just how DNA is. You're not going to get a double-stranded DNA molecule that's not 5 to 3'. They won't match. And to try to put in words that you might understand, the tension, if you try to put it any other way, there'd be a lot of tension. Like overstretching a rubber band, and then you let it go, what happens? It just snaps back to the way it was, right? And so that's what's going to happen with these DNA molecules. If you try to fit them in an opposite or a weird direction, the hydrogen bonding won't let it move in that direction. It's like trying to turn something that's been Velcroed together. It's very difficult, and they tend to break. So, we're moving on. There's an enzyme that makes DNA. So you have this molecule that's double-stranded, right? It's double-stranded and it's a double helix, right? So 2 molecules. Everybody agree? Everybody okay with that?
Yeah.
2 molecules and it's double-stranded. There it is. And in order to make a copy of it, we know it has to make 1 new strand and 1 old strand. We have to open it up. It has to open up. So instead of being this way, it actually starts off doing its double helix, but then it opens up. Starts off doing double helix, and then it opens up. So this is called the replication bubble. And the reason it's called the replication bubble is in part that it when you look at it, it looks like a what? It looks like a bubble, see? See how it opened up here? And this open region is called the replication bubble. Something had to open up that unzipped the helix, right? Something had to open it up. And the enzyme that opened up.
That's semi-conservative.
Say that again?
Is that semi-conservative?
Yeah, that's right. We're working on, we know this from many biochemical experiments, everything I'm telling you. But we know from the experiment Messelson Stahl that this is semi-conservative. That's what led us to this investigation. So the very first thing that has to happen is something has to break up the helix. What do you think we're going to call that enzyme that breaks the helix up? A protein that breaks the helix up. Huh?
He just said
ACE is how we end it. And the beginning is what it does.
RNA.
Close, very close. It's called, to make the replication, unhelix it, to unwind it, right? We called it helicases, right?
Oh, I had it right.
Yeah, you were close. She said dihelicase or something like that, but it's very. No, you're very good. That was a great guess. It was absolutely. She's saying it makes absolute sense, right? Helicase breaks up the helix. Not difficult, right? So the replication bubble is made by helicase. At least the beginning of it's made by helicase. Then then something has to come along. And there's all kinds of other enzymes that are involved in this, but we're going to keep it simple. I'm trying to keep it very simple. I'm going to go ahead and turn this sideways here. And so what we have, if I can make this work, okay. So we're going to need one of these, huh? We'll have to wait on the quiz tomorrow because I need to finish this, right? I don't want to leave this halfway open. So what we're going to do is we have to take this DNA molecule and we have to then copy each strand. Is that right? So if you don't mind, I'm going to move forward. I'm going to leave this as is. I'm only going to show you the replication bubble. Can I do that?
Yeah.
Just half the replication bubble. So in fact, I'm not even going to switch slides. I'm just going to delete that. And we're going to just then look at this half. All right. Let's look at, I could even move this over here because this is the only part I'm interested in right there. All right, so if this side here is 3', Follow this along. Follow this along. What is this one over here?
3 prime.
5 prime. Very good. And if this is 3 prime here, what's this?
5 prime.
5 prime. Excellent.
Wait.
Yeah, go ahead.
The other one, it was both one of them on the same side.
Oh, which one?
They had, you have 5 prime on like the same side. It was 5 prime, 5 prime.
Yeah,
Well, let me pause this. So here we have 3 prime and 5 prime, right? And so yes, in the other example, you ended up with 5 on this side and 5 on this side, but you have to follow the actual molecule. So had we followed the molecule, there would have been one more loop. You might have found it on the same side, but it's all about where this molecule is, right? One side is 5 prime, the other side has to be what? 3 prime. Always the opposite ends, anti-parallel. Okay, so there's a code here. I'm not writing it. Everybody understands that there's AT, GC, AT, right? All the nucleotides are here. All the 5 prime, the ribose sugar with the nitrogenous bases, right? This is a phosphate backbone. Write that down, phosphate backbone. It's a sugar phosphate backbone is what we call this. Sugar. Why do we call it a sugar phosphate backbone?
Because that's what it's made out of.
That's right, that's what it's made out of.
Sugar.
Corny is good if you remember, as long as you remember, because that's all that counts. All right, so sugar phosphate backbone is this sugar phosphate. It's a complete repetitive sequence of sugar and phosphate from 3 prime to 5 prime and from 5 prime to 3 prime. Here's a problem. Well, let me tell you first what is the name of the. What do you think is the name of the molecule that? What do you think is the name of the molecule that makes DNA? DNA. Boom. Just that simple. It's actually called DNA. It's not DNA section. It's DNA poly. You did not say it. DNA polymerase.
Did not just hear me say all of that?
I did not.
I heard it.
Polymerase.
I said that word.
Dylan, doesn't that break up?
I love that people said DNAs. So DNAs breaks up. DNAs breaks up DNA, right? What makes the polymer of DNA? DNA polymerase. See? So DNAs breaks it up. DNA polymerase makes it. And really simple, kind of, and we try to make it simple in science. If you pay attention, actually, most of the stuff is pretty straightforward. But the hard part is that everything builds on itself. If you forget or didn't learn one piece, you lose it as you move forward. Is it 57? Oh, too bad. Well, we'll finish this tomorrow. All right. I think we're moving along pretty quickly. But you need to please study this stuff tonight. Please do the practice problems and know this for tomorrow. So when we continue, we're not lost. I emailed you this picture yesterday. I expect you to review it because for the exam, this is a summary of DNA replication. Here's everything you need to know about DNA replication. Does everybody understand that? Yeah. All right. So who did that? Who looked at this and who can identify what's going on?
I looked at
Who can look at it and tell me what they think is going on?
I know.
Go ahead, Johnny.
So they unwrap the DNA from the chromosome, and then they put it inside the replication thing, and that splits up the thing. And like from there, splits up the 2 strands. From there, since like each one has to match with a certain one, since it's only like. They just match it up with the one that's supposed to match up. It's like a zipper. And like they unzip it.
You know what? Why don't you let him talk? Because I think I understand where he's going with this, and I'm not disagreeing. Go ahead, someone. Go ahead, Johnny.
They unzip it, and then like it's going to like another zipper. Each strand is going to another zipper. It's like putting the things back in from the.
Okay, go ahead, next.
And the colorful things, there's that's like a key. The adenine and the thymine are all connected throughout the DNA, and the cysteine and guanine are.
Right.
And they split apart right there.
Right. Okay, so I like where you're going, especially since we haven't even discussed it yet, so I'm glad that some people have looked at it and tried to figure out what it is. And what you're dealing with, you see that this is a chromosome. Do you see the centromere right there?
Yeah.
So this is one of the sister chromatids, the other one's on the other side. Or actually, this is the one that's getting, when this is done, you're going to build a sister chromatid that you're going to attach to that centromere. Does that make sense? Yeah.
Why is it free nucleus?
Well, the nucleotides, remember, are these monomers. They're kind of floating around. When they say free nucleotides, that means they're not attached. So you should write that down. You should make sure that you understand that free nucleotides means these are nucleotides that these enzymes are going to use to build the new strand. Does that make sense? And so if you remember back at the Messelson-Stahl experiment, oh, thank you. I have some more copies of the testing schedule before you guys leave. And according to the Messelson-Stahl experiment, as you see, as if you remember, they would have added these nucleotides, which have what they would have had the nitrogen 14, right? Nitrogenous bases. These are the nitrogenous bases. These little yellow sticks here, you see they represent the different nitrogenous bases. The purple part represents the sugar phosphate backbone, right, that we just looked at. So what happens is that this enzyme named DNA polymerase, and I told you about it yesterday, didn't I? This DNA polymerase is going to take these free nucleotides and do what with them?
Match it with the old.
Match it with the old strand and then connect them together chemically, all right, covalently bond them. So this enzyme, what it's doing is it's connecting these nitrogenous bases to one another in a new strand. It's creating the new strand. But of course, it's doing more than that. It's matching it's just like Johnny said, they're matching the old strand. So where you see an A here, that's going to put what?
A T.
A T. When you see a T here, it's going to, I think this is actually a G, and this one is a C. That doesn't seem to have matched, but let's see, this one would be a C, this one should be a G, yeah. CG here, and here's
Green
CG.
and yellow go together. Green
Right, so you know that it's going to be red with blue and yellow and green, right? So that's what should be going on here, and it seems it is, except this one seems a little weird. It's going to be T with an A. A-T, no, that's right. It's A is going to go with T, yeah. So in any case, this new strand is being made, the old one's being read, and the DNA polymerase is what's doing it, right? It's reading it, it's attaching them, it's covalently bonding them into a new strand. It's doing some other stuff too. It's checking for mistakes. So if there's a mistake, if it puts a G with a C, it makes sure that it corrects the mistake in the new strand. So it has editing capability as well.
So it's like a factory.
It is exactly these, and that's something that you should know is that enzymes, and these are all enzymes, right, that we're talking about. What they do is they make reactions easier These reactions would not happen. The reactions happen so slowly without the enzyme that you could say they would not, would not, do not happen without the enzyme. So what I'm saying is that the reaction, you might find nucleotides finding themselves and bonding to each other on their own, but they just would take so long it wouldn't make a difference to life, because life happens in minutes and seconds and hours and days. Some of these reactions would take 1,000 years to happen on their own, all right, to any great extent. Alvin's son, you gotta wake yourself up. Either stand up, go put water in your face, whatever it is. You look like you're having a hard time breathing. You okay? You all right? Okay. Because your eyes are red, you're breathing through your mouth. You seem kind of out of it. All right. Yeah.
Okay, so the enzyme is like a restriction enzyme, right?
Right. We'll talk about restriction enzymes in a minute. You've learned about restriction enzymes in this Udesipatus class. It's really good. What restriction enzymes do is they cut DNA up in specific sections, and we found these enzymes in bacteria. The thing is that we'll talk about that a little in a minute, but these enzymes do something different. They're taking look at what they're doing. They're taking the free nucleotides. This specific enzyme, DNA polymerase, is taking the enzymes, the free nucleotides, and attaching them, checking them, fixing them. But there's some issues that we have to deal with. These reactions, you would not expect free nucleotides to make a DNA molecule on their own under our conditions, under the conditions of the Earth right now, in anywhere close to the amount of time that life needs it to happen. Okay? So enzymes, what they do is they speed a reaction up. So make sure you know that. Enzymes, as I will test that. Enzymes speed the reaction. They don't, let me write this down first. They're not used up in the reaction, so make sure you know that. They're not used up.
So they can go for everything.
Hold on. They're not used, enzymes are not used up in the reaction. They don't get involved in the reaction. They just make a space where it can happen easier. Okay? So have you seen those dating sites?
Blackpeoplemeet.com.
Yeah, whatever. Shh. Oh God, if you guys don't calm down, I don't know what got into you this morning. But when people come together, people come together on their own, don't they?
Yeah.
But what a website that allows people to get together is creating an environment that allows people to get together easier and faster, right? They have these places you can go, these restaurants you can go, and you can meet. There are people line up to get in there, and they call it speed dating, where you pick a number, and you sit down with someone, have a cocktail. You got like 10 minutes to meet them and talk to them, and then you move on to another person. Everybody has a card, and you exchange cards. And what is that environment doing?
Speeding up the process.
Speeding up the process. That's what the enzyme does. That's it. It's exactly like a catalyst. It is a catalyst. It's a biological catalyst. So that's good. Where did you learn about a catalyst? In chemistry? Oh, that's right. You're in chemistry. Huh? So for those of you that have taken chemistry, for sure, it's a catalyst. It's a biological catalyst. It speeds up the reaction. The way it does it, and I'm not sure they're going to test this, but I'll go ahead and tell you, is every chemical reaction has needs what's called activation energy. All right, so this is energy. It goes up in this direction, and this is time.
That's like a graph. Oh.
So this is a graph, yeah. So this part here, this is called activation energy. Can you read that? Do you need me to kind of...
I can read it.
Can't see it. Would you hear?
All right, so activation energy, and the thing about activation energy is you know what it is. You've seen it happen. Everybody has. So you're out in the woods. Anybody started a fire at all at either the woods or in a barbecue or a stove, right? No. So there's some people that haven't started a fire at all? Yeah, so everybody has. So when you take, all right, so you're going to start a fire. I'm going to go and get, oh wow, you're going to force, go to the bathroom. Don't interrupt me again. I've been really nice this morning, don't you agree?
Yeah.
You need to stop. So anyways, you have, let's say I want to start a fire. I go and get a can of gasoline or some coal that I put it on the table. I got a fire, right?
Put it on the table.
I put it, what do I have to do?
You have to ignite it. You have to activate it.
I have to activate, I have to light, you have to spark, give a spark, right? That lighter, that little bit of energy is energy, okay? And that little bit of energy is known as the activation energy. So almost every reaction requires some, well, that's not true. A lot of reactions require a little bit of energy to start. That's called an activation energy. Reactions don't just happen without any energy to begin with. Did you write that down? Because you're going to need to know that. What enzymes do, I'm about to draw it, and catalysts do the same thing, those of you that are taking chemistry. What enzymes do is they do this to a reaction. What do they just do?
Make it faster.
They made it faster. How?
And less power.
They're right. They lowered the activation energy.
So it takes less, and they do more.
That's right. They do exactly what that speed dating place does, right? When you go on a date, it takes a lot of energy. You're going to have to find the girl, right, or boy. You're going to have to let them know that you like them in some way. Buy them flowers, write them a letter, ask them out. You got to ask them out. You got to go and act. A lot of work goes into getting to know that other person. Guys, can we not argue details? This is an example. It's a metaphor for you to use. All right? You're just going to keep talking in the background. It's not going to be able to do it until I kick you out. I'm sorry. So, the speed dating site, it takes all that other extra work you would spend all that money and energy on, right? And what does it do? It makes it go faster by putting you together in the same room and making it a lot less energy for you to get up and go to another table than to get out of a relationship and go find another girl or boy and start a new relationship. This speeds up the process by making it easier. Does that make sense?
Yeah.
So that's what this does in a way. It's creating an environment where the molecules can find each other, and it's easier, and it lines them up in the right angles so the bonds can happen. So it requires less energy to make it happen, and so that's what increases the speed. Okay, that is the activation energy, and that's what we're talking about here. That's what these enzymes are doing. So again, I'm going to put in this picture that again you've been emailed, so you should know. Oops, that's not it.
Moldings.
That was for another project. So here you have. So everybody understands then that these enzymes, what they're doing is they're lowering the activation energy. Now there's different enzymes here, and there's some problems. I told you that these molecules are opposite one another, right?
Yeah.
Right. Well, the problem is that you have well, it's not a problem. I'll go ahead and say it's a problem. The problem is that DNA polymerase, the molecule that puts them together, okay, that puts these two molecules together, makes the new strand. It can only read the DNA reads the DNA only from 3' to 5'. It reads 3' to 5', and it makes it 5' to 3'.
So what if it changes?
It doesn't. It can't change. It can't do anything else. It's the only thing DNA polymerase can do.
Is that not good?
Well, think about it. What that means is that let's say you have a replication bubble. I'm going to miniaturize this for a second. This is why it doesn't. This is why it requires a number of enzymes to make DNA, because you need a DNA. If this is 3 or 3' end, then DNA polymerase. Let's make it look like this. Okay, DNA poly can come in and make a new strand, right? No problem. But this is a 5'. How can it do it there?
You have to get the other one on the other side.
Great.
It'd have to start over here. You'd think it has to start over here, but that's not how it works. What happens is that you don't get one single replication bubble. You get a replication bubble here, and then another one here, and then you might have another stretch, and then you have another replication bubble here, I'm glad that there's some kids having conversations. I hope that some of your parents come and ask me why you didn't get a good grade on this final. I'm going to use this video as an example. I'm going to tell them, listen to this. Listen to me beg your children to please listen and pay attention as I explain this really difficult process, and listen to me say to them, without mentioning their name or trying to embarrass them, that they are not listening and they're not paying attention when I'm trying to explain it. That only a few of us in the class, about two-thirds of the class, are actually engaged and having conversation, while your children were in the back talking and getting distracted by one another and refusing to listen. What a good conversation I'm going to have with your parents at that point. I am uploading this to YouTube. I will share this with your folks if they come in or ask me. And I get no other way of communicating to you that you can't do this. So anyways, DNA polymerase, it can only read from one side, guys. It can only read from one side. It can only read the 3' end. And it starts to make the 5' end. So this is an issue. This is a really big problem for DNA polymerase. Of course, there's a solution, right?
I thought it was anti-predator. How's that a problem?
Well, it's not a problem for one half the DNA. But it's a problem for the opposite side, right? What don't you understand about that? Tell me.
No, because you guys said that that's a problem, and I thought that's what they're supposed to do since it's anti-parallel.
Okay.
Right. But I'm confused on why you said it's a problem.
Go ahead. Keep. I want to hear more. You're confused about why it's a problem.
Yeah. Because you said it's anti-parallel, and that's what it's supposed to be.
Right. I see what you're saying. Yeah, it has to be that way. There's no other way it can be. The problem is that this enzyme can only read one of the strands.
Oh.
How do you make a copy of this side then?
Oh, because it just...
Because you got to make two, right? You see you're making one here. So then you got to make one of this one. This one is fine because this is the 3' end over here. This one's not okay because this is the 5' end. How is this going to make, this is the 5' end here.
So it doesn't even look...
This is the 5, so it can't, so DNA polymerase can't latch onto that, right?
Start from the end.
So let's talk about what the solution is. Obviously there's a solution because it happens, but the solution is kind of a little complicated. So first of all, the first thing you're going to need to do, by the way, to any DNA polymerase is you need a primer. You need to lay down a primer. So this is kind of a summary. So let me draw this out here. Before you have to open up, the first thing you do, you got this DNA, right? What's the first thing you have to do? You have to open it up. You have to stop having this twisting, right? You have to remember what I told you, if you had braids and we had enemies on the other side of a border, right? But we wanted to send a message to one of our spies, and we put the message inside your braid. What would the spy have to do in order to read it? Unwind it to read it, right? And so DNA is all wound up, right? And in order to read the message that's here, there's a message here, right? There's a message here. In order to read it and make a copy of it, you have to you have to unwind it. Does that make sense?
Yeah.
All right, so that's the first enzyme that you need. You need this enzyme called helicase. And there's a bunch of them, but I'm just going to mention a couple. Helicase. That makes sense, right?
Yeah.
Helicase is the one that unwinds the helix. Yeah.
So you know how you call it double helix? For it to open up both of them, do you have to, does the amount matter of how many helicases you have? So,
Good question. Well, I guess if I understand your question, the only time you need a helicase in this DNA molecule is right here, right? You need a helicase right here. And this helicase is unwinding. Now there's other things that get involved. There's little proteins that stick on this so that they don't rewind, because if you unwind it, it'll bounce back and rewind by itself, right? Rewind into a double helix. But what happens is the little proteins stick on and they stop them from sticking together. But we're not going to worry about those proteins, all right? I'm leaving a lot of stuff out. I'm trying to give you just the basics, okay? So what happens is helicase unwinds it. Now, the first thing that has to happen is that you need a primer, all right? So you need a primer that's going to come in, and we have this enzyme called, guess what? Real hard word. It's primase. You need a primer, and a primer is just a little bit of, it's an RNA, it's a little bit of RNA, and I'm going to go ahead and use pink. It's RNA. It'll get another enzyme will come in and change the RNA into DNA. It'll get rid of the RNA and put in DNA.
I thought DNA comes first.
Well, that's not good.
Like it's supposed to be.
Well, that's right. You're right. It's not that simple, is it? So this primase, what it does is it reads the message, okay, on DNA, and then it puts on an RNA equivalent. So if this is AT, I'm going to reuse it. Let me use a different color.
Don't you use U in this one?
Yes, that's right. Good.
A-U?
You got it. That's right. So if it's ATGCAT, right? Over here it's going to be what? UACGUA, right? That's what's here in this RNA, but it's just stuck on there. It's not meant for, do you understand that so far? Can I erase that?
Yeah.
Okay, so that's that RNA, and what that does, that RNA primase does, is it's like a landing strip. It's like a landing, a plane needs to land, right? You need an airport for a plane to land, right? You can't just land a plane in the middle of the forest, right? Because you need a long strip of flat land. Does that make sense?
Yeah.
So this RNA ends up being the landing strip for RNA polymerase. I'm going to use a different color. So RNA polymerase comes in, and that's my bad. DNA poly, okay? So DNA polymerase can land on that, all right? It can land on that because this is a free 3' end. What it does is it makes a 3' end, and because that's the 3' end, it's able to then land and start reading, no problem. Here's your 5' on this side, and it leaves a free 3' end on this side. Is that okay?
Yep.
All right. So you need that landing strip for DNA polymerase. But then you got a problem because over here, this is a 5' end. If you made an RNA equivalent here, what would this be?
3.
This would be a 3, and then what would this be here? A 5. And 5 doesn't work for DNA polymerase. It needs a 3' end, doesn't it?
Yep.
Wait.
So that's that. Let me rewind. Look, pay attention real close because this is kind of tricky. So if we try, if this here. No, how can I do this? Can I just undo this and just?
No, let's do all of them.
Wait, pause it.
Yeah, I'll pause it. Hold on. Let's say this is a 3' end. I'm opening up, drawing the DNA in a different way. And then this is a 5' end. Let's say it's open, all right? And then, of course, over here, these are hydrogen bonded, okay? These are hydrogen bonded. Helicase is still opening up the double helix. Let's assume that, okay? And you have this, you're still going to have a primase coming in and landing and making a primer. And let's make the primer blue. And let's say on the 3' end it makes a primer. So this is 5' here, right? This is 3'. Now DNA can land on that, right? Here's your landing strip, right? This is made of RNA. It's made by an enzyme that we call primase. Made by primase. It's a primer. A primer is just something that's a beginning. It's a beginning of the message, all right? So this is the landing strip. Now DNA polymerase can come in, no problem,
Turn it into DNA.
and start to make DNA polymerase.
Oh, that makes sense.
And because there's your 3' end, right? It's no problem. The DNA polymerase is no problem making more DNA, and I'm going to use a different color so we can make the DNA a different color, different from the white. Let's use green for the DNA of the new strand, okay? All right, so DNA polymerase, of course you'll have the DNA polymerase will be made. Right? And that DNA is going to be made by this enzyme here, right? Everybody okay with that?
Yep.
This is called the leading strand.
Wait, look.
This is the leading strand. There's no problem in making the leading strand because it's just DNA polymerase landing on the 3' end of a primer and reading through. If you have to go to the restroom, go ahead. Make sure you use your pass.
5'. Or there is not.
So, yeah, there is a name for the other molecule. Now, the problem we have is this one here. This one here starts with a 5'. If we, pay attention, because I'm going to do this subtle change here. Right, we will, you're right, but let's not jump ahead. If we tried to make a primer here, do you see this? Does everybody see this? Because I'm drawing this, I'm pointing to something. Does everybody see this? Because some people aren't even looking at the board, right? If we did this 3' end here, and then we have a 5' end over here, DNA polymerase cannot land here, right? Because it's 5' end, so it doesn't help us. It doesn't help us. So what does the system do? What have we evolved to be able to do then? That's right. We start on this side. We go ahead and do primase lands. It lands on this side. If this is the 5' end, what's this end? 3'. So if it's 3' here, what's going to be over here? 5'. And then on this side of the blue molecule is going to be what? 3'. Now you got a 3' end. Now DNA polymerase can land, right? And now DNA polymerase can land.
I have a question.
Yeah, hold on. DNA polymerase can land and make a big, a nice little strip of DNA. Unfortunately, it can't do that for too long. So what happens is that you have to have another fragment of RNA, another primer, and then another DNA polymerase has to land. Could be the same one, right? And it's going to keep doing that. It's going to make these little strips, these little pieces of DNA that will have to be ligated together or put together. So what you're going to end up with in the end is you're going to end up with kind of this that has a little bit of blue, and a lot of green, just a straight kind of green. And then you're going to have the other molecule, the lagging strand, is going to have little blue, and it's going to have a lot of green. And what these things here, these are called, and this is an important term, these little fragments on the lagging, this is called the lagging strand.
That makes sense.
Yeah. Lagging strand, and this lagging strand is going to have these things that are really weird named, but it's actually kind of cool. Okazaki fragments.
Who's gonna remember that?
I will.
It's a really easy to remember because it's the weirdest name you ever heard. Okazaki fragment is named after the professor, the researcher that discovered it. It's weird to your ears because it's a Japanese word, and his name was Okazaki, and he discovered it, and so we call it. We honor him by naming it after him. And so you have this Okazaki fragment, right, that's going to make the second copy.
And it just keeps doing it.
And it keeps doing it that way, but it's because it's doing it slower, we call it the lagging strand, okay? And because this one's faster, we call this the leading strand.
But they're both making the DNA.
They're both making DNA, it's just the lagging strand has to work harder because it has to do it in pieces and parts. And then the one last enzyme I want to talk to you about is ligase. Ligase is what's going to ligate these together, bind these together, and get rid of the Okazaki fragments. So what you just went through, guys, I know it's time to go, so just listen. What you just went through is understanding this. I want to run through this with you one time before you leave, please. So this is the leading strand. This is just an RNA polymerase. Laid down a primer. This DNA polymerase will read it straight through, no problem.
Right.
Here's the lagging strand. DNA polymerase is going to read in the opposite direction. You see how this was making DNA this way. This was making DNA this way. It's going to make DNA in this direction in pieces and parts. You're going to have something called an Okazaki fragment. What is this thing that's unraveling? It's unwinding this. What's it called? Helicase, DNA polymerase. What's going to take the Okazaki fragments and bind them together?
Enzymes.
Ligase. So know those terms, know those terms, know those terms. They will be tested, and we're done with DNA replication. That's it. That's all you have to know.
When are we gonna take the quiz?
Your quiz will be tomorrow. Sorry, I had to finish this. And this morning, people came in with too much energy.